To begin, let us acknowledge that Queen's University is situated on traditional Anishinaabe and Haudenosaunee territory. We are grateful to be able to live, learn, and play on these lands. We thank the Indigenous peoples of this area for their care of their land for thousands of years, and we hope to honor and respect them as we hold our virtual event today. I'd also like to take a moment to thank everyone personally for calling in today for our presentation. Our speakers this afternoon are Hannah Caird, Dr. Aaron Fraser, Stefan Iwezawa, and Quinn Stewart, who will be co-presenting on the topic Trends in Tick Surveillance and Tick-Borne Diseases in British Columbia. Hannah is an epidemiologist at the British Columbia Centre for Disease Control, or BCCDC for short, where she has worked on the enteric and zoonotic and vector-borne team. She completed her Master's of Public Health with a concentration in public health at Simon Fraser University in 2020 and joined the BCCDC as a data analyst in November 2020 before transitioning to an epidemiologist role in August of 2021. Dr. Fraser is a public health veterinarian at BCCDC and a clinical assistant professor in the School of Population and Public Health at the University of British Columbia. She is a graduate of the University of Guelph and has over 20 years experience as an epidemiologist, public health veterinarian, researcher, and executive director. She co-founded Veterinarians Without Borders Canada and was the organization's first executive director. She has worked across Canada internationally with interdisciplinary multicultural teams to develop programs and projects that address public and health issues from zoonotic and vector-borne disease surveillance, prevention, detection, and control to wildlife health, food security, and factors that affect livelihoods. Her current work at the BCCDC focuses on surveillance of zoonotic diseases and foodborne pathogens, antimicrobial resistance in companion animals, and climate change and tick-borne diseases. Stefan is a project coordinator for the Ticks and Climate Change 3 West project at the BCCDC and the Center for Coastal Health. Over the past 15 years, he has worked in the field of veterinary epidemiology to identify and understand the interactions of human, animal, environmental health. Quinn is an epidemiologist on the enteric, zoonotic, and vector-borne diseases team at the BCCDC. She has a Bachelor of Sciences in Biology and Human Geography and a Master's of Public Health from the University of British Columbia. We are going to have the research team present, and then we will open for questions from the audience. You can ask your questions by entering into the chat box, or you can raise your hand icon when the opportunity arises later on today. Please help us welcome Hannah, Dr. Fraser, Stefan, and Quinn to the podium.
Wonderful. Thank you for this opportunity. And I'm great to see some colleagues from BCCDC are also joining and colleagues from some of our other areas of work. So I'm just going to also acknowledge where I live and work. So I'm joining from the traditional unceded territory of the Lekwungen people, specifically the Songhees and Esquimalt First Nation. So also just wanting us to take a moment to recognize the First Nations communities that we are joining from today, and recognizing they have a historical and relationship to land that certainly continues to this day. So just to give you an overview of what we're planning to cover today, so we're going to turn it over to Stefan in a minute, and he's going to give us a background of our tick species of concern in BC and a little bit about their biology. I'm going to then just give you an overview of our tick surveillance program in the province. From there, Hannah is going to take the reins and present some of our passive tick surveillance results. And then Stefan will come back on and give us some results around our eTick program for the province and the active tick surveillance results. Then I'm going to just touch on our climate change and tick-borne disease project that just is in the wrap-up stages, linking in with some of the climate change pieces. And then Quinn is going to give us an overview of our Lyme disease trends. And at the end of all that, Stefan is going to run a pop quiz to test your tick IQ. So without further ado, I'm going to turn it over to Stefan and just tell me when to advance the slides when you're ready.
Wonderful. Yeah, so I'm just going to run over some ticks of concern in British Columbia, and I'll jump into the next slide. Aaron, thanks. Perfect. So I guess the two big ones that we're going to be looking at today and of our concern for humans and companion animals is Ixodes pacificus, also known as the Western black-legged tick, and dermacenter species. Mostly Andersoni is what we have in British Columbia, but we do get the odd variabilis, also known as the Rocky Mountain wood tick and the American dog tick, respectively. I just thought I'd point out on the left-hand side there, you can see there's a larva who's got the 6 little legs, and then in the middle, the nymph, and then finally an adult male. And just to give you a rough idea of size, just down below there, my unmanicured thumbnail, you can see is a picture of a larvae. So those are actually the same 2 guys there. And just to give you a rough idea of how small they are and how difficult it is to spot them. So this is actually a field collection that I did. Give you a rough idea of how big they are. And if you look at the nymph, which is likely to bite you and get away with it, down on the bottom right hand side there, just get a rough distribution map of where you're going to be finding Ixodes ticks, Ixodes, and along with the dermacenter species. You can see there's basically just the south coast and Vancouver Island that you're going to be running across those Ixodes pacificus ticks. You could jump to the next slide, Aaron. Perfect. And for just the life cycle of Ixodes pacificus, generally it's a 3-year life cycle. It can happen in 2, but it's generally a 3, starting in kind of the 1 where the eggs are laid, and you can see it turns into once they hatch, they turn into larvae. And if you see just to the right there, I don't know if you can see my cursor, probably not, but there is a lizard there, basically just above spring and summer there on the right hand side. I just want to point that out because it is one of the favorite kind of the happy meals of larvae when it comes to food choices take that blood meal from fall off and go into the leaf litter, and it's going to molt over the year and come into year number 2 where they're going to hatch as nymphs. And when those nymphs have hatched, they're going to be looking for larger hosts. You're looking at your deer, your humans, your raccoons, your foxes. Once they've taken that blood meal, they're going to fall off again and molt and turn into adults, at which point the female is going to be looking for another host to take a blood meal in order to lay those eggs once she's met up with an adult male. And then they're going to be laying those eggs, and the cycle continues. So one interesting note is that a lot of times people will ask me when I'm in the field, oh, is this year a bad tick year? And they're not similar to mosquitoes where you can have a spout of hot weather like we're having currently in British Columbia. You have that water, the females are going to be laying lots of eggs, and there's going to be lots of mosquitoes forming. It really comes down to the last couple of years. Has it been a really good couple of years, let's say 3 years, in terms of when you're going to have a big bout of ticks appearing all of a sudden? Wonderful. Next slide. And I mentioned before, chalk one up for the lizards. The lizards are one of the favorite hosts for nymphs. And you can just see on the left there, the western fence lizard, which is found down more in California, southern Washington, Oregon. And you could just see that little spot there on the side of its neck where I believe it's a larva or a nymph has attached and taken a blood meal. And the one interesting aspect of this, it's in the literature showing that the western fence lizard has a protein in its blood. Now, when the tick, which is positive for Lyme disease, has got the Borrelia burgdorferi in its gut, when it attaches to that western fence lizard, takes that blood meal, that protein actually neutralizes, kills the Borrelia burgdorferi in the gut. So when that tick falls off, it is no longer positive for Lyme. And that's one of the attributes and one of the reasons why we believe that Ixodes pacificus has such a low rate of Lyme disease when it comes to testing. Luckily enough, we have a northwestern fence lizard, and you can see just the distribution. It's on the right-hand side there in green. And this is maybe one of the reasons why we have that low rates up in British Columbia too, because we have this northwestern fence lizard that could have the same protein in its blood, basically counteracting the Borrelia burgdorferi. So a positive tick that does feed on one of these lizards could fall off and become negative after the fact. I think that's next slide, if you could, Aaron. I think that's on to you now.
Great, thanks, Stefan. So I just wanted to give you an overview of our tick surveillance program in BC. This has evolved over time. So the blue components are really the mainstay of our tick surveillance program. So on the passive side, we have 2 components there. We have direct submissions from primarily physicians, veterinarians, some of the health authorities submit ticks directly to our BCCDC public health lab. where up until current day, they have tested any Ixodes types of Ixodes ticks for the agent of Lyme disease, Borrelia burgdorferi. They are in the process of launching several new tests, multiplex tests, to be able to capture some of the other, or to look for some of the other tick-borne pathogens, including Anaplasma, Babesia microti, Ehrlichia, and Rickettsia. So that's an exciting new development that's been coming over the last couple of years. The other piece of our passive tick surveillance, which Stefan will go into more detail about, is around the eTick program. So we joined, BC joined the eTick program back in spring 2021. And this has been a really nice asset for us. So members of healthcare providers, but members of the public, across the board can submit pictures of ticks for identification, and then they're provided with information about that tick species that's found, and whether it's a concern for tick-borne pathogens, for example, some instructions about what to do if it's found on your pet, if it's found on yourself, those types of things. And then on the active tick surveillance side, there have been several research projects historically. I think one of my colleagues, Sunny Mack, is on the line here. He's been very involved with our colleagues at the lab, doing some active tick surveillance at key sites in BC. But currently, since 2019, we've partnered with the Canadian Lyme Disease Research Network to develop the active surveillance sentinel sites, which Stefan will explain in more detail on some of those results. That's in partnership with the National Microbiology Lab, where they conduct the pathogen testing, and that information is shared back to our team in BC. And then in orange, we've got some different special projects that we work on to enhance our intelligence around tick species and tick-borne pathogens in BC. So a couple examples of that, we have a partnership with Merck Animal Health, where ticks that are collected from animals are tested for pathogens of concern. We had a first version of that, which ended in the ticks, any Ixodes ticks were tested for Borrelia burgdorferi. And then currently we have a sort of 2.0 version of that project, where now ticks that are identified as Ixodes species using the eTick program, we request those ticks to the BC Centre for Disease Control Lab, where they are tested with these new multiplex tests. So that's just getting up and running, this just starting in late January this year. And then we've had some other partnerships with, for example, our Ministry of Forest Wildlife Health Program. They collect ticks from different wildlife species, ungulates, moose in particular, and getting those identified through some of our collaborators. And so that just gives you a quick overview of how our system in BC is set up. And I'm going to turn it now over to Hannah to describe some of the passive tick surveillance data.
Thanks, Erin. So firstly, I'm going to touch on the tick submissions from human and animal hosts to the BCCDC Public Health Laboratory from 2002 to 2021. So during those years, 13,881 ticks were found on humans and submitted to the BCCDC Public Health Lab from locations throughout the province. And of these, the majority were Ixodes species or dermacenter species, so 37% were dermacenter. And of Ixodes ticks with an identifiable species, the most common were Ixodes pacificus, 92%, and then followed by Ixodes angustus, 7%, Ixodes ricinus, 0.2%, and Ixodes scapularis, 0.2%. So Ixodes pacificus and Ixodes angustus ticks are known vectors of Lyme disease, which causes Borrelia burgdorferi. In addition, Ixodes pacificus is also known to carry babesiosis. Ixodes ricinus species is a species of European tick known to carry many pathogens, including Lyme disease, anaplasmosis, and Ixodes scapularis is a known vector for Lyme disease, anaplasmosis, and babesiosis, as well as Powassan virus. Next slide, please. So incidence rates of Ixodes species in tick submissions in humans have been increasing from 2002 to 2021 in Vancouver Coastal Health and Vancouver Island Health Authority specifically, so that more coastal region of British Columbia. And that began in 2013, where it remained stable in other regions in British Columbia. And the highest rates of Ixodes species were found in Vancouver Island Health, followed by Vancouver Coastal Health. Next slide, please. So the majority of ticks that were sampled by the BCCDC Public Health Lab were, in descending order, Ixodes pacificus, Dermacenter andersoni, and Ixodes angustus. Dermacenter constitute the majority of human tick submissions from interior health and northern health specifically. And in interior health, the incidence rate of Dermacenter andersoni has been increasing since 2009, which you can see in this figure here. And in northern health, the incidence rate of Dermacenter andersoni has been increasing since 2011. So peaks of those incidence rates of Dermacenter andersoni and Ixodes pacificus have submissions peaked in 2015/2016, as well as in 2019. And in Fraser Health, Vancouver Coastal Health and Vancouver Island Health Authority, Ixodes pacificus was the most commonly submitted tick species from human samples. And the incidence rate of Ixodes pacificus submissions from Fraser Health has fluctuated over time, ranging between 2 to 4 ticks per 100,000 population per year. And then in Vancouver Coastal and Vancouver Island Health Authority, the incidence rate of Ixodes pacificus has been increasing since 2012. Next slide, please. So looking at the seasonality of tick submissions to the BCCDC Public Health Lab by region, the majority of ticks were submitted between March and June. In Northern Health, the northern part of the province, submissions are low all year round, with slight peaks in the number of ticks submitted in the months of April and July. And there is a slight increase in the number of ticks submitted in Vancouver Island Health Authority in the month of November, but still low overall. Next slide, please. So looking at the number of Ixodes tick submissions and the proportion of them that tested positive for Borrelia burgdorferi, the causative agent of Lyme disease, from 2002 to 2019, we see that the percent of Ixodes pacificus and Ixodes scapularis tick specimens can be highly variable, and the peaks in percent positivity vary in a pattern occurring every 2 to 3 years, mirroring the life cycle of ticks. So we've excluded submissions for 2020 in this slide, since there were very few ticks testing positive that year. And that was followed by a particularly high year in 2021. And we see submissions of about 1,300 ticks per year and 500 to 800 Ixodes ticks per year. And peak submissions are in April through June. And that's all for me. I'll hand it back to Erin.
Great, thanks, Hannah. And we're going to advance and turn it back over to Stefan.
Perfect. Yeah, so I'll just describe some eTick and some quadrant data that we've been looking at over the last couple of years in BC. So if you want to jump to the next slide, Erin. So of course, I'm sure everyone's familiar with eTick, but just in case, free, easy, and fun. If you haven't been there already, please do check out etick.ca. I know there was a presentation earlier on that by Jad, but basically it's a public platform for image-based identification and population monitoring of ticks in Canada. And I thought I'd show you just a couple of quick screenshots on the right-hand side there. There is a free app that you can download from the App Store or the Google Play Store. Basically, you open it up, you hit the red submit button. The next slide over on the right there, you can see I've submitted a couple of quick photos of the ticks that I've caught. And then on the bottom left there, it just lists some details in terms of where you picked up that tick, where you're located. On the right, you can actually drop a pin. This was actually sampling in a local public park just down the road from me. So you can drop a pin where you think you caught it. And then on the right-hand side there, they actually keep track of your submissions. So they let you know whether or not it's been vetted. So basically, usually within 24 to 48 hours, an eTick technician in your province will get back to you saying, hey, this is the type of tick you got, and this is what you should be looking out for. So it's a great program, and I really appreciate all the general public and practitioners and everyone just taking part in it. As you can see, since 2017, around 36,000 ticks have been submitted online. That number's probably increased because this is an older slide, but it's really great to see that the Canadians are taking part in the project. Next slide. So just looking at the data, specifically 2021 versus 2022 in British Columbia, you can see that the number of submissions in 2021 are quite low, 348. Reason being, we were in the midst of the pandemic. There wasn't a lot of push or publication of what was going on in terms of eTick. It was a kind of a soft launch. So definitely a lot less submissions 2021 versus 2022. We did up our game. As you can see in 2021, only 55% of those ticks were valid submissions. So you get invalid submissions, not a tick, it's a spider, or some people actually submitting Google images saying, tick, and then they just drop the picture and they're saying, this is what I saw. So unfortunately, those are not valid. 2022, 1,300, just over 1,300 ticks submitted, 994, 74% were valid, which is great to see because the national average, of course, on the right-hand side, they're around 76%. So we're definitely upping our game. Next slide, please. Tick submissions by month, which is really kind of cool. Hannah just showed us the submissions that we see at the lab, and we see that kind of same curve where you have March, April, May, June, a larger amount of ticks being submitted compared to the rest of the year. And again, 2021 being a lot less compared to 2022. And minuscule, but it could be there on the right-hand side. In November, you do see that second little bump going on of tick submissions in the province to eTick. Next slide, please. If we're looking at breakdown of what those ticks are coming off of, what kind of hosts, so free in the environment, off humans or off of animals. Of course, humans leading the pack in terms of being the host for those ticks. And of course, the numbers again being more in 2022. Kind of an interesting breakdown. 2021, we only had the two, cats and dogs. Dogs, of course, less likely to groom themselves, so they're not going to be catching those ticks, munching them or spitting them out. 2022, we definitely have a wider variety of hosts, bovine, cats, dog, goat, horse, moose, Fifi the rabbit there submitting a tick for us. So great to see, a wider spread of hosts in 2022 versus 2021. Next slide. Submissions off humans by host range, you can see it's pretty even across the board there. The one thing I did want to point out is that 0 to 10 age group there on the far left-hand side. In both years, 2021 and 2022, it's much higher than what the rest of the age classes are. The reason being is kids are more likely to leave the footpath when they're going down in trails and stuff like that. They're running off and picking up pine cones and doing this and picking up sticks and of course picking up ticks at the same time. So it just really goes to show that you definitely have to do those tick checks, and specifically for those 0 to 10 age group. It's been found in the literature too that kids are more likely to be the hosts because they wander off path and are going to be brushing up against things, and that's when you're going to be collecting those ticks off the foliage and stuff like that. So that low-lying grass and bush and stuff like that. So make sure to check your kids for ticks and your dogs because they're more likely to have those ticks once you get back to your car. Next slide, please. This is actually maps off of eTick. So we're looking at all the submissions in 2021 on the left, and then all the submissions on 2022 on the right. And I just wanted to point out on the left there, you can kind of see it's very bottom of the province, kind of southern side of British Columbia versus on the right-hand side, you can see in that turquoise box there that I've got where those 2021 submissions are and where the 2022 submissions. So definitely seeing more submissions further up north, which is we're really trying to encourage because there is a lack of data. Less people, of course, and that's probably why there's less submissions to eTick, but every tick from up north is a valuable tick because it helps us to fill in the blanks in terms of where these ticks are located and kind of prevalence of number of ticks. Next slide, please. Moving on to Clydron. So I've been doing some active sampling for Clydron. This is a map of where the sample regions, the sentinel regions were in 2022. So there was around 35 regions, each one of those around 5 sites, around 200 sites were sampled last year in 2022 for Clydron across Canada, which is always just mind-boggling how many, how many sites and how many hours and stuff like that, because it does take a fair bit of time to go and sample each one of those sites and identify them. Next slide, please. So the sites that we did sampling in British Columbia, we had a big focus on kind of the southern Vancouver Island and British Columbia, because that's where Ixodes ticks are found. So we have the sampling region in red there, kind of the northern mid Vancouver Island, southern Vancouver Island in blue, the Vancouver region there in green. And then in 2022 was the first year we did some sampling in the Okanagan, where mostly dermacenter ticks are going to be found, but there have been submissions to eTick and ticks off of companion animals and humans that are Ixodes. So we're really curious to see how that's changing and getting some baseline numbers for that. Next slide, please. So this is the breakdown of results of the ticks collected in 2022. We had 119 ticks collected, the majority of which Ixodes pacificus nymphs. You can see there 49%, and then kind of a sprinkling of all the other different kind of age classes. Ixodes angustus there, of course, and the dermacenter, of course, coming from the interior. So great to see that. Of those 119, they're only going to be testing. So once identified, they're going to be testing Ixodes ticks, and they're going to be testing nymphs and adults because the larvae haven't taken a blood meal yet and shouldn't be carrying Borrelia burgdorferi. So next slide. So I think it was 82 to 84 ticks actually qualified for testing. So once we did that testing, you can see the sample sites on the left there, and then this kind of a zoom in on where we had our positives. So over in Vancouver, we had 2 Babesia macrotei positive ticks, 2 different parks. There was no co-infection of any of these. These are all individual ticks with one pathogen inside them. So Babesia otocolii, which can also cause babesiosis. I think it was a paper just came out in 2021 where they did some blood testing of humans in Ontario showing that, yeah, you can cause babesiosis from otocolii. So 4 of those in a Gulf island. And then finally on the bottom there in Victoria, 1 site we had a tick that was positive for Borrelia burgdorferi, which is of course the causative agent of Lyme disease, and 2 more ticks that were positive for Anaplasma phagocytophilum, which can cause anaplasmosis. Next slide, please. Oh, there you go. And it's on to you, Erin.
Great. Excellent, thanks. So I just wanted to highlight a few, this project that is just wrapping up. So this climate change and tick-borne disease project, it's a Western Canada initiative with Alberta and Saskatchewan, and it was funded by the Public Health Agency of Canada under their Infectious Disease and Climate Change Fund. So we essentially had 3 primary goals with many different components in that, and I won't go into a lot of detail, but just our 3 main goals were really to improve our cross-sectoral detection, monitoring, and surveillance capacity for ticks and tick-borne diseases across Western Canada, looking in more detail around data, acquiring surveillance data around Anaplasma, Ehrlichia, and Borrelia, and Babesia, which is not listed here. We also had a goal around increasing prediction and response capacity for health professionals and at-risk communities. And I've got a couple of slides, but my colleague, who I believe is on the line, Dr. Cork, did present on some of this work at the session just this past Friday. And then the overarching goal was really to just strengthen our One Health capacity to address climate-driven diseases. So hopefully translatable to other types of climate-driven diseases, mosquito-borne diseases, other tick-borne pathogens, and so on. So we had a structure in terms of a secretariat group represented by members of each of the provinces, and had a series of working groups, one around tick surveillance, another one around modeling, and then knowledge translation. So some of the pieces, we've got some manuscripts in the works or already published. and doing presentations such as this and sharing what we've learned through this process. So one of the slides, which I believe, I imagine that Dr. Cork showed on Friday, but I'm just going to showcase it here in terms of some modeling work done with on Ixodes pacificus, using sort of a, I guess you call middle of the road climate prediction. So the green is in the current habitat suitability for the Ixodes pacificus ticks, and the blue is highlighting future areas where the habitat will be highly suitable for Ixodes pacificus ticks. So sort of builds off of that south coast where we have the abundance of Ixodes pacificus ticks now, but highlighting other areas up, for example, in Haida Gwaii, in the Terrace River valleys up in northern BC, where habitats may become quite suitable. And then also those Okanagan interior BC valleys where we may get further expansion of the ticks. So just to highlight this modeling work was done at this RCP, the representative concentration pathway 4.5, where I've got that arrow, but recognizing this is conservative. Unfortunately, globally, we're on track for the RCP 8.5. So I think this is a conservative estimate of where our habitat suitability will go under future climate scenarios. And some other work that Dr. Cork and her team at the University of Calgary conducted and have produced this manuscript is around Asian longhorn tick. The point here is just highlighting how BC is highly suitable for this tick. While we do not have this tick here, I just want to emphasize it is not present. It's one that we really want to keep an eye on. We know this tick is it's definitely a One Health issue. It's a concern for animal health and human health. These ticks are very, they're generalists, and they can reproduce asexually and so can rapidly propagate. We know that in the eastern US, there are, last I heard, 17, 18 states that have found this tick. So it's important that we're monitoring for it and know what to look for. So getting the systems in place to be able to detect this tick should it arrive in BC, either by gradual migration or a species introduction, dropping off a bird through livestock movement and so on. And so there's been some great resources done by Public Health Ontario. The Canadian Animal Health Surveillance System has also put out a really nice preparedness document for Asian longhorn tick. And without, okay, so that's my last slide. And I'm going to now turn it over to you, Quinn, and she's going to talk about our Lyme disease trends in BC.
Great, thank you. Yes, so I'm going to be talking about some of the human Lyme disease trends we've been seeing in BC. So Lyme disease has been provincially reportable in British Columbia since 1994 and nationally notifiable in Canada since 2009. So from this figure here, it shows the number of confirmed Lyme disease cases per year since 2006, when the first case of Lyme disease was reported in British Columbia. And the blue there are the confirmed cases, that were inquired within the province, and those in gray are cases that were reported amongst BC residents, but were acquired outside of British Columbia. And as you can see, a large portion of our Lyme disease cases that were reported in BC were acquired through travel outside of the province, and that includes both travel within Canada, so travel to another province, or as well as international travel. And human cases of Lyme disease in British Columbia tend to follow a cyclical pattern where they peak every 4 to 5 years. And the annual number of cases reported range from 5 cases per year to 39 per year. And this past year in 2022, we saw a total of 10 cases of Lyme disease, 5 that were locally acquired and 5 that were travel related. And this translates for an incidence rate of 0.1 locally acquired confirmed cases per 100,000 population. Next slide, please, Erin. So this figure from the 2019 preliminary annual report on Lyme disease by the Public Health Agency of Canada really puts into perspective the relative burden of Lyme disease across Canada. And as you can see there, the Western provinces, including British Columbia, tend to record relatively fewer cases of Lyme disease compared to the Eastern provinces. And in 2019, BC reported 14 cases. And in that same year, Ontario reported over 1,000 cases, so much fewer cases in British Columbia. And to better put in context the magnitude, over the last 29 years since Lyme disease has been a reportable condition in BC, we have seen a total of 213 cases in BC. Next slide, please, Erin. And this figure here really illustrates the age and sex distribution of our locally acquired confirmed Lyme disease cases in BC. The majority of cases, 57%, for males, which was 62 cases in total, and 43% for female. And in addition, most of our cases tend to be adults within the 45 to 49-year-old age group, followed by adults in the 60 to 64-year-old age group. So a little bit of an older adult population here. Next slide, please. Just ending off with the seasonality of our locally acquired confirmed cases of Lyme in British Columbia, we tend to see a big increase beginning around this time in April and May, with the number of cases generally peaking in August. However, we do see some smaller peaks that occur in November, February, and April. Now, I hope you've all been paying attention because I think we're going to get hit by a quiz by Stefan. So hope you took some notes or kept some things in mind.
Great. Thanks, Quinn. I'm just going to stop sharing and turn it over to you, Stefan, for
Sounds good.
our quiz.
Dun, dun, dun. Quiz time. So if you can visit slido.com or scan the QR code there on your smartphone, and you should be able to bring up, hopefully everyone can see that quiz there. And I'll give people just a couple of seconds to be able to jump into full screen so you can see all the whole screen there. Maybe I'll just count down to 5 in my head. And hopefully everyone has a chance to pop over to slido.com and enter in the number or scan it on your smartphone, their QR code there below. So our first question, spot the tick. Which one of the following is a tick? Which one of the following is a tick? Let's see if people have been paying attention. I think that was one of the first slides up there. So feel free to put your answer in the Slido. Oh, someone's writing on there, Nina. Perfect, circling it E. And I'll also show the results. I'll just give people a couple seconds. 3, 2, 1, and let's see how our audience polled today. So 91% saying E. Oh, that's great. Oh, that's good to see. Yeah, the correct answer is E. E is the tick in the group of not ticks. Next question, how do ticks catch a host? How do ticks catch a host? They grab onto you when you brush by them. They fall or jump onto them from trees. You get them from your father's side of the family. At least that's what my mom always tells me. And they create a profile on plenty of ticks, which is open to all ticks. Dermacenter, Ixodes, soft tick, hard ticks. You take your pick of ticks. They're open to it all. I'll give the group just a couple of seconds there. 3, 2, 1, and let's see where we're going. Oh, perfect. That's great to see. Great to see that everyone knows that they grab onto you when you brush by them. So next question, applying a small amount of perfume, gasoline, Vaseline on a tick is the best way to remove it. True or false? Applying a small amount of perfume, gasoline, Vaseline on a tick is the best way to remove it. I've also heard of people taking matches. That's another one I've heard when I'm out in the bush there collecting ticks. Let's check out the results, see how our polls. Perfect, wonderful to see. Yeah, the best way to remove a tick, of course, is by using a pair of tweezers or a tick removal tool, going to the base of the skin where the mouth parts are and just gently pulling up, not twisting, not yanking, and just waiting till that tick pops off, trying to make sure you grab the mouth parts there and not squeezing too hard is the big thing too. Wonderful. And we're jumping to the next question, question number 4. Can all ticks transmit Borrelia burgdorferi, Lyme disease? Can all ticks transmit Borrelia burgdorferi or Lyme disease, the causative agent there? And let's see how the audience is doing. Oops. I'm going to stop annotating. There we go, and show the results. Perfect. No, yes, and it's Ixodes ticks. Interesting enough, when you go along to the interior of British Columbia, we see a lot of those dermacenter. I get a lot of questions saying, "Oh, isn't it Lyme disease?" And then just having to notify the public, no, it's not. When it comes to dermacenter ticks, it's only the Ixodes ticks that can transmit Lyme disease. Specifically, Ixodes pacificus and Angustus are the ones that are going to be biting and could potentially have Lyme disease. What % of lab tested ticks in BC are found to be positive for Borrelia burgdorferi, the causative agent of Lyme disease? What % of lab, this is a toughie, lab tested ticks in BC are found to be positive? A couple more seconds. Let's see what the audience says. Ooh, there we go. So it's 1 to 3%, less than 1%, 6 to 7, or 8 to 10. And the correct answer is less than 1%. Less than 1% of ticks in British Columbia that are tested, historically speaking, are positive for Borrelia burgdorferi. That was a tough question. Oh, this is my favorite. What is eTick? A hip new food delivery app catering to the parasitologists and all of us, or the next generation of Apple Watch that is designed for everyone who ticks along in their daily lives. Abbreviation of the word etiquette when texting. Should be smaller than that, maybe even smaller than that, ET or something like that. An online platform for image-based tick identification and population monitoring in Canada. Or finally, last but not least, a bricks and mortar boutique located at 1832 Sherbrooke Street, West, Montreal. Or is that Etiquette Boutique? I'm always get those two mixed up. I think that's enough time. Let's see the results. Perfect. Everyone knows, of course, eTick is an online platform for image-based tick identification and population monitoring in Canada. Although Etiquette Boutique is a real thing, honest, Google it, it'll come up. Question number 7. Can you always feel when a tick bites you? Can you always feel when a tick bites you? I don't know how many people have had ticks bite them in the past. No, I'm glad to see not that many hands up from the audience. Let's check those results. False, which is correct, not true. It is correct. You can't always feel when ticks bite you, and I can vouch for that. I've found multiple ticks on me that I didn't know bit me in the first place. Question number 8. Ticks are only active in the summer. Ticks are only active in the summer. Let's see what the polling tells us. False, then that is correct again. Yeah, ticks can be active, especially in British Columbia on the island. You can have them year round. So we do get ticks submitted to eTick and to the lab year round when it comes to ticks. So definitely not just the summer. This one might be a bit tough. What Ixodes tick is currently is not currently established in BC versus other parts of Canada? What Ixodes tick is not currently established in BC? Is it Ixodes angustus, Ixodes scapularis, Ixodes pacificus, and Ixodes or Ixodes artuleus? This one might be a bit tough. Okay, and I'll show the results. And that is true. It's Ixodes scapularis. Ixodes scapularis is currently not established in British Columbia. Thank goodness, one less tick in the province for sure. Last but not least, final question. Ixodes pacificus is mainly found where in British Columbia? Is it up north, northern British Columbia? Is it mostly along the south coast of British Columbia? Is it the southern interior? Or like a great big blanket in wintertime, evenly distributed throughout to keep us all snug and warm? And I'll just wait 3, 2, 1. Perfect. Yeah, mostly along the south coast of BC. So it looks like our audience have had their Wheaties this morning and are doing very well. And that's the last question. So I'll stop sharing. And I think we're heading over to the Q&A question portion of the event, I believe.
Thank you, Dr. Fraser, Stefan, Hannah, and Quid for your engaging discussion presentation today, as well as the quiz. That's always fun to do. Thank you so much. I'm looking to the chat to see. I don't see any actual questions typed into the chat right now, so I'm going to move it over to the audience. Oh, we do see someone entering a chat question, so I will read that right now and see. It looks like a comment. Let me just read it a little bit longer. Is there any comment that you would like to? Yes, thank you. Respond to the comment at all? You've had a chance to take a look at the comment. while you're reading it over?
Yeah, no specific comment from me.
Okay,
No.
perfect. Thank you. Is there any questions from the audience? Feel free to raise your hand icon and we can go to your question. We have a question in the chat box. Any future plans to test Ixodes pacificus for Borrelia neomyto in BC? This can infect larvae, nymph, and adult ticks.
Yeah, that is a good question. I feel like my lab colleagues would need to respond to that, and I don't think anyone is on. I know that there has been several detections of. Sorry, is Sunny on the line? I don't know if he.
Hi, yes, I am here. I do have a friend that's been talking about it. I'm not sure who's added to his testing procedures.
Sorry, I couldn't.
Yeah, it was a bit crackly. Aaron, from my understanding of the multiplex text that has been developed at the BCCDC does cover Borrelia species, and one of those being Miyamoti. So I'm pretty sure if there were detections going on, they would be able to catch that with the new multiplex test that is coming out, or is out and is currently being tested.
Yeah, it's just in the final validation stage. So I think we're using it for some special projects, but I think it's not. Yeah, but there are plans to activate that once the validation steps are done.
Thank you. Do we have any other questions from the audience? Don't be shy. Please feel free to unmic and ask your question, or you can raise your hand icon if you have a question. I know you guys are out busy in the fields right now. Is there any indication, any change this year at all on surveillance, how it's going this year compared to last year, or is it a little bit too early to say?
I think it's in terms of numbers and stuff like that. Yeah, we're almost halfway through the sampling sites, the 20 sites that we have in British Columbia. The number of ticks that we're finding, I'm pretty much on par. There's a couple of sites that we didn't catch ticks last year that we're catching them this year. So pretty even in terms of what's going on so far. Definitely more dermacenter ticks that I found in the interior this year compared to last year. But of course, this is just the second year that we've been sampling at this site. So tough to say whether or not that's an anomaly or something that's new and changing.
Thank you. Dr. Kadur, please go ahead and ask your question. We can't hear you, Dr. Kadur. Do you have it? It looks like you're unmiked. Yes, we cannot hear you at all. Yes, can you maybe type your question in and I'll be happy to read it off. We'll just give him a second. In the meantime, while he's entering his question, does anybody else have a question from the audience? Okay, the question is, how about the lizard protein as a target for Lyme disease therapy?
I don't know that any of us would be able to answer that because that's not our area of.
Okay.
Yeah.
And are you aware of any research being done elsewhere, perhaps that not involving your research laboratory?
I am not. I wonder if Dr. Cork has any information on that. Do you, Susan?
There's quite a lot of work going on in the US. A number of laboratories have presented at the vector-borne disease workshops that have been held across the US and various new project initiatives, but I don't think the actual data has been published yet. But I think people are looking into whether or not it'll help us understand therapeutic options in the future.
Thank you. Any other questions from the audience? I'll how about any new projects that you might be starting up with your research team in the coming year?
That's a good question. So yeah, the funding for the current initiative, the climate change and tick-borne diseases, was not renewed. So yeah, a little bit back to the drawing board in terms of the funding source to continue some of that work. There's been some amazing collaborations established amongst our Western Canada group. So, you know, we certainly are looking to explore some of the build on the modeling work and information sharing around our tick surveillance practices across the provinces, but no firm plans at this point. And then in BC, we've got some special projects, as I mentioned, with the Merck Animal Health. So that's just getting just got up and running in January. So that's the current one. And then ongoing work at the lab, which I can't speak to in detail. I'm not a lab person, but around that final validation pieces for the multiplex tests.
Thank you. One last chance from the audience. Now's your chance. You don't usually get everybody together in one room. So if you have a question, let me just check the chat. Yeah. Yep, just a lot of thanks for your presentation today. I do want to thank Dr. Fraser, Stefan, Hannah, and Quinn for presenting today on their topic. And I do want to remind everybody, I'm just going to share my screen to show you that our presentations do continue tomorrow. Our next presenter is Dr. Amir Kadur, who will be presenting on long-term antibiotics for Lyme disease with persistent symptoms, a pilot project with 468 patients. That presentation is tomorrow at noon, 12 o'clock Eastern time. So hopefully you can join us. I also want to remind everybody about our challenge. Remember to wear green, take a photo and share it with us to help spread awareness of Lyme disease and tick-borne diseases. If you don't like your photo taken like me, you can still take pictures of your beautiful artwork, whether it's poetry, sketches, drawings, that express how you feel Lyme disease and tick-borne disease awareness month is all about. Take those photos, submit them to Clyder@gmail along with your name and your email, and you'll be automatically entered into a draw for one of 4 $25 Starbucks e-gift cards. And we will be doing our draw on our last day of our presentation at 1 o'clock on May 31st. And finally, just to let you know that TickNet Canada will be having its inaugural scientific symposium in Toronto, Ontario, October 24th and 25th. The planning committee is almost finished finalizing all the details, and we hope to open up registration abstract submission in early June. We hope to have some more details for you towards the end of the month, so look for that as well. Again, I just want to take this opportunity again to thank the BCCDC research team for your presentation today. Thank everybody for attending today's presentation. I hope you have a great rest of the day and hope to catch you as we continue our presentations this week, tomorrow, as well as Wednesday and Thursday. Take care, everybody. Have a great day.
Thanks.
Thanks. Bye for now.
Thank you.